Jude Fitzgibbon was awarded BA in genetics from Trinity College Dublin in 1989 and his PhD from the University of London in 1993 for a project involving the positional cloning of tuberous sclerosis gene on 9q34. Following postdoctoral positions at the Institute of Ophthalmology and the Institute of Child Health, he was appointed lecturer in the Centre for Hemato-Oncology in our Cancer Institute in 2002. He was promoted to senior lecturer in 2004 and then again to reader in hematological malignancies in 2009. In 2013, he was made our professor in personalised cancer medicine. Professor Fitzgibbon's research focuses upon the genomic evolution of follicular lymphoma and in particular the specific tumour-initiating changes in DNA and histone methylation that appear to accompany the shift from normal to malignant B cells. He has current programme grant funding of approximately £3 million, including awards from CRUK for a research programme in personalised epigenetic therapy in follicular lymphoma, and from LLR for a research programme in familial leukemia. Professor Fitzgibbon, as with most of our professors, is in much demand as an external speaker, and his CV lists invitation lectures in just the last 12 months to the 2014 Epi Congress in London, the Department of Human Genetics in Hanover, Germany, European Association of Hematopathology in Istanbul, Ruijin Hospital, Shanghai Institute of Hematology in China, Biennial Childhood Leukemia Symposium in Prague, Biotech Research and Innovation Centre, University of Copenhagen, and last, but certainly not least, for a pint of science at Bethnal Green Working Men's Club on the subject of epigenetics. I won't share with you which of those venues I have been to in the past 12 months, but the number and breadth of his presentation suggests that we are in for a treat this evening. So, Professor Fitzgibbon, may I invite you to present your inaugural lecture, enigmatically entitled, "It's a Long to Tipperary."
Thank you all so very, very much for coming. I'd like to welcome everybody to the Queen Mary University, Barts London Medical School, and more importantly, the Barts Cancer Institute. I can't tell you what an honour it is for me to be presenting an inaugural lecture here, but more so to be actually a professor in the Centre of Hemato-Oncology, because I joined here 17 years ago. And so what I'd like to do today is to give you a story about who I am, where I'm from. Also the journey from Tipperary, Southern Ireland, to the third floor of the John Vane Science Building. And lastly, and critically more importantly, I want to thank so many people for helping me along the way. And it's great to see so many people that I did my PhD with, Kay here, Sue Povey and Jonathan Wolfe, who were my supervisors 25 long years ago. And hopefully you can kind of get a flavour of our research along the way. Because whether we like it or not, I think academic research at the present time is really hard. And there's a certain amount of luck, there's a certain amount of playing the game, or in my case, it took me a hell of a long time to actually realise there was a game to be played. But more so is if people don't like what you're doing, if your peers don't appreciate it, if you can't translate your excitement for your research across to everybody else, then really you're doomed. And I've been very fortunate to have a lot of hands. I've had a lot of hands to actually work and help me along the way. And it's that I think, first of all, though, I'd like to really give a flavour of who I am and where I'm from, because when you're small and in Ireland, and I think it's probably wherever you are, people will say to you, who are you and who's your father? Because in a way that defines so much who you actually are. So if anybody wants to send a letter or a cheque in the post in any shape or form, they know that it's very easy to write a letter to me because I'm Jude, I'm Fitzgibbon, I'm from Capel White, and I'm from County Tipperary, and that's the address. It's very simple. Or in Gaelic, I'm Jude Mockibun, Capel na Fuitach, County Tipperary. This is the school that I went to. and my father actually went to. He's 93, so clearly not here today. But he's 93, and that's the school, a village of about 300 people. And this is Joe and Jude Fitzgibbon. Every man is born as many men and dies a single one, because that means we have a lot of choices to make. We all have potential to be whoever we want to be. I'd be very pleased to be anything like my father. He's a wonderful man. Joe has several different kind of quotations. He's very good at quotes. He likes quotes like I do. So when I told him I was going to be a professor, the first thing he said was that when I finished my PhD 20 years ago, I told him, I'm going to be a professor someday, Joe, but you won't be around because you'll be dead then. He had a little twinkle in his eye. He said, I stuck around. I like analogies. I like comparing things with different things, and you'll see that in my talk. And so what I kind of do is I make these analogies, and Joe's impression is that my analogies are anomalies. That's what he says a lot. He also says one other thing. He says, you have to be true to yourself. And he also says something else. He said, now that I'm 94, I stop asking people, what are they doing, or what do they have, or where did they go on holidays? What he's keen on to know is, are you happy with what you have? Because they're the things that are important. So if I could be anything like him, and he would say I'm not anything like him, I'd be like, my mother is a most wonderful lady. Kind, very gentle. She was a nurse. She was a midwife. She trained in Dublin and in Limerick. She went to Liverpool and she went to New York. She came back at around 30 years of age to kind of settle down in her family. 4 boys. So I'm a twin, and I'm the eldest. I'm the second actually. Dan is the eldest. But Mammy is wonderful. Now Joe wouldn't travel, but Mammy would, and she'd be sitting in here along with brother Joseph and my daughter Anne. And I'd like, because she's so close to us, because she died of motor neuron disease about 7 years ago, I just want to give a kind of a little flavour of kind of the interaction that we'd have just before I'd come out and she'd sit down. Because what she would say is, she would first of all say, Jude, you understand, I won't understand a word that you're going to say here. And I'd be able to say, well, actually, half the people in this audience have said that to me at some stage in the past. So you're not, but in particular, Professor Hart has said that to me on numerous occasions, and I really feel the success for what it is over the last few years is really down to him. The only person that Mammy would have heard about is Professor Lister. So the next thing she would say is, she would say, where's Andrew? And I'd point over there to Andrew, and she'd have a look. She'd say, he doesn't look too bad. I said, well, that was 7 years ago, things have changed. And then she'd say, who's that elegant lady sitting beside him? And I'd say, well, that's Sarah. You should meet Sarah later on. And then because she's an Irish mother, and she's probably the same mother as everybody else has, she'd begin a bit more confident, and then she would say something like, does this mean you'll have a job, Jude, for the rest of your time here? And I go, ooh. I said, the person that asked about that is Professor Lemoyne, but Nick isn't around today, but he's been really helpful to me. He's been so helpful to me, Nick Lemoyne. But you said what Nick would say is, as long as Jude keeps going, Mrs. Fitzgibbon, the way he's going at the present time, he'll be fine. And 2 other people I'd like to kind of pick out is Delphine and Lynn, because I think, although they don't directly do the research, the support that they've provided in relationship to my working day over a long period of time has been huge. The next thing I want to finish on in relationship to kind of that backwards spiral is, I love team sports, because every young fella in a village, you're playing with your schoolmates, you're playing for your village team. The people who are coming are your parents, but they're also the parents of all the other people, and you have this tradition, you have this love, this expectation that you're playing for your village team. And I really have tried in the last 5 or 6 years to try and instill that into the different people that work with me, because in a way, it is about multidisciplinary research. It is about working as a team. And we have to win together, we lose together. And victories definitely are sweeter working as a team. I have to say, I don't believe that we mourn together and makes things easier. I don't think it does. I think failure still hurts the very same as if you're by yourself or with a team. But that team part has been, to see it in front of groups of people that we're working very hard in hematological malignancy has been amazingly wonderful to me, and I think it's been an inspiration. And I think it's a reflection on how we're doing just at the present time, because the people have engaged in that specific kind of question. This is Jude, believe it or not. Now, it's very important to realize Jude was born in 1967. What happened in 1968? The Beatles released Hey Jude. I just want to, it's very important to realize. Now, I know Mom went and worked in Liverpool, but I still feel that it's very important to say that. I went to Trinity and did natural sciences and specialized in genetics. And the Irish American Society do a kind of a scholarship program, and I was very lucky. I think I came 8 out of 15 in my class, and the 8th person, the first 8 got to go to upstate New York, to Buffalo. Quite a big change from Tipperary to go to upstate New York, but I stayed there for 3 months. We worked on genetic mapping around the locus on the X chromosome using mouse backcross. And I think it immediately made me aware, do you know something, Jude? You can do this and you can be good at it. And I think that's terribly important for young people starting off today, to actually realize that, you know, the people I'm working with are the best, and I can be as good, if not better, than them. And I think that's what happened at that stage for me. Then I went to the Galton Laboratory for 4 years with Jonathan Wolfe and Sue Povey. I didn't realize how wonderful the training I actually got was. And I'm going to talk about that in a little bit more detail. Then I went to Morton Ophthalmology for 3 years. You can see the cancer theme swinging over and back here very nicely. I did a very unsuccessful postdoc at Gosh for about 12 months, decided I had to leave. And then in 1997, I moved to the Center for Hematology, the previous Center for Medical Oncology. I stayed there for 17 years. And I think when you come as a young postdoc, at a kind of a crossroads in your life, 2 small kids, and wondering, am I going to be able to get traction and survive, essentially? I think that when I look back at the 17 years, I have a lot to be grateful for, a lot of different people who helped me over that time. Technology has changed a lot, hasn't it? God. So when I did my first PCRs, and this is the wonder about science back in the day, is that you sit with the 3 PCR machines, or the 3 water baths. You take your floaty, you have to find a floaty to start with, and you keep moving them over with a stopwatch. That's how you did your PCRs. Gave you plenty of time to think. Made you realize what the buffer contained, because you actually made the buffer. It's 1.5 millimolar magnesium. That's what you need. And then you have to find a pH meter. Oh, they were tricky to find. But Duran bottles were pretty tricky in the Galton to find at the time. Things have changed immensely. And this is how now we should be looking at the human genome, because I think it'll make a huge difference in how we can play and can have fun with the genetics to try and understand cancer and lots of other diseases. Because this is a 25,000 football stadium, but it's 25,000 genes, and they all look very different. They're all responding. They're all responding to the environment, and then they get very excited, and you can regulate their expression together. Because they're not genes, are they? They're genes with clothes on, and the clothes are your epigenetics. And it's the epigenetics that is controlling the expression. And now what we've got an opportunity to do is to actually play with the genome in a way we could never do before, because we can sequence all 25,000 genes simultaneously. We can listen to their expression. We were talking about microRNAs and long-coding RNAs. We can do the works. And what we need is well-annotated clinical series, lots of money and clever minds to actually underpin and decipher the basis of the individual cancers that we're interested in. I want to go back to my PhD, tuberous sclerosis. So essentially, I arrived in Jonathan's lab. Hasn't changed very much actually. This is Kay Taylor. She was the senior PhD student in the lab. She was an immense help. She was hugely, hugely sympathetic and understanding to the poor Irish guy who just got off the boat. And has, on and off, we've talked a lot over the last 17 years. And I think that she was a wonderful help and a wonderful kind person in the laboratory. This is Nick Hornigold. I was trying to remember what exactly Nick did. But Nick came in every day with the Telegraph under his hand, and between experiments, he would read the Telegraph. And I'd be on the next bench. And the one thing that Nick introduced me to was test match special. Because cricket was, oh God, come on, you're swinging a bat. Nick would listen, I'd listen. And first of all, England were losing all the time against Australia. Because it was only during the summer months we had these 5 tests. But test match special and the guys and the team and the camaraderie and the commentary box, hugely important. I was making something called the radiation hybrids. When I think about it, it was a fantastic PhD. I dreamed of cloning TSC1, the gene on chromosome 9q. That was my dream. I expected to clone it. And we had no idea of human sequence. We had a region around the ABO locus, arginine succinate synthetase, ABL locus. That was the kind of region we thought that the linkage was. There could have been hundreds of genes, because we had no idea the number of genes even in the region. But we made these irradiation hybrids, southern blots. I just think anybody who sees a southern blot under a UV and actually can see the cream of the DNA digested beautifully, if they don't like science, then, or they don't get excited looking at it, then you say, oh, maybe you should go and do something else. We need heroes. So I'm an Irish kind of person who's soft on the outside and hard inside. Okay, so every so often I get a little bit emotional. Sue Povey is my hero. I could say about Sue so many things. Fantastic scientist. Wonderful to the people that she worked with, Mary Wynn, Steve Jeremiah, lots of other people. Huge contribution to the fields of alpha-1 antitrypsin, TSC1, TSC2, and now the Human Genome Mapping Project. But Sue Povey is something else. When you went to talk to Sue, you were the only person in that room. She could focus in on you, smile, talk, think, support you, guide you. And not for a moment did you feel that there was something more important than what she had to say to you. Unique, because we're so busy these days. We're so busy trying to get our list done. Always had time. And I can't tell you, even if it's 17 years ago, or 25 years ago since the PhD, how important Sue Povey has been to me. David Hunt is where I did my next postdoc. David was probably less important, because this is as fun as David ever was. Jim, Jeffrey, and Neil here are going to disagree. And we worked on I decided to take a different route in genetics and evolution. And it was a lovely idea, because there's a lake in Lake Baikal called Siberia. Lake in Siberia called Lake Baikal. It's highly oxygenated throughout, and it's very, very deep. And the fish live, and they've evolved from the same species. And so we were able to sequence their genomes and actually work out which amino acids were linked with the absorption. Because the different wavelengths of light correlated with these amino acid shifts. So it was a nice evolutionary path, a nice story. But what was key to me is when I was there, there was 5 or 6 fellas. There was Binoy, there was Kamal, there was Jeffrey, there was Jim, and there was Neil. They knew when you were running a gel what you were looking at, when you were developing an autorad, what you were looking at, and there would be a debate and a discussion. And we never set the world on lights, scientifically at the time in any shape or form, but we loved what we did. And they mean a lot to me as individuals. Gosh, it was the right move, and I would say to anybody, you make the move, it doesn't work out, you leave. I left after about 15 months. Maybe I should have left a bit earlier. Didn't work out. I was working, but it was interesting. I was working in acute myeloid leukemia. I was working in the Department of Developmental Biology Unit. The head of the department was a wonderful guy called Peter Thurgood. I'm not sure does anybody know Peter Thurgood, but I used to walk with Peter from King's Cross to Gosh. Maybe once a week we'd have a chat. Peter fell off a mountain and died in Switzerland. And you begin to realize, if we're going to do something, do it today. Decide what you want to achieve, and then go ahead and try and get that done. Because we just don't know where the next mountain is going to happen for each of us individually. That's the way I looked at it. Now I have 3 stories to tell. One is on patients and engagement, one is on leukemia, and one is on lymphoma, and then I'll be finished. And I might overrun by a few minutes. Raj Gupta gave me my job here. I was terribly pleased he did, because I was at that crossroads. Postdoc, 2 postdocs in, not really sure where I'm going next. Raj was great. He did something for me. Professor Malpass told me when we were doing some research work 17 years ago in the tissue culture, patients with multiple myeloma, and he said, Jude, if you ask them for your hand, a patient would, because they're so worried about themselves, they want to try and give as much as they can to try and improve treatment, and improve treatment for themselves, but if not themselves, somebody else. Raj Gupta took me to clinic. So I've seen thousands of patients, but they've come in little tubes. I've only seen about 20 patients in the flesh in clinic. And Raj took to me, and the first patient I met was a 65-year-old woman who had hormonal replacement therapy, because Raj told me, because she looked marvelous. And her paraprotein levels were going up. She was there with her husband, and she was frightened because her daughter was getting married in Australia in 12 months' time. She didn't care what Raj gave her, what the long-term consequences might be. All she wanted was to be able to wear her hat in 12 months. And it changed the way I look at things, because you kind of think of living and dying, but you also have to think about all these things that we're here for. To further graduation, for the Holy Communion, for Arsenal to actually win a cup at long last, for Fulham to be relegated, whatever it's going to be. You have to think in relationship to the hat and what people want for themselves. And this is the first patient I met, and I was inspired because, first of all, Raj gave me a job, but he also gave me an understanding of why I want to work in hematology, why I want to work in cancer research, and why I think it's so important to be driving towards better outcomes. And it was rather wonderful then, because this year, Cancer Research UK gave me something called the Flame of Hope Award. About 50 a year, so it's not bad. It's not one, but it reminded me of 3 things. First of all, I met people who give a lot of money to Cancer Research UK. And they asked me 3 things. This person asked me one big question, which was, what would you do with the money if we gave you the money now? What would you try and do? And I told him, I wish I could take more risks. I wish I could fail, because we're not allowed to fail anymore. We have to succeed, or we have to write like we succeed. You can't write back to the grant body and say, you give us 500K, we worked really hard, but it didn't really work out well this time. But can you give us some more, please? But that's what we should be trying to do more of. That really is what we should. I gave a talk to the Cancer Research UK shop workers society or shop workers in Greater London. And they spent an hour talking about, because I went early, they spent an hour talking about how to make the perfect frontage of the shop, because then they could get an extra £25. That hit me. And probably what hit me most was I went out to Southend, Simon's neck of the woods, to the lymphoma support group for the LLR. And there was a guy who had relapsed diffuse large B-cell lymphoma. My mother would say he was a strong, good-looking man. And we talked about the research, we talked about what we're doing. He came to me afterwards, he shook my hand, he said, "Thank you so much for treating me like an adult," because he felt we had a chat about his disease, what we were trying to do and where we were going. These kind of things mean a lot to me, and I'm so glad to see that within the BCI we do so much on this engagement, because I think it's very important. These are the past fellows and PhD students. I could talk individually about all they do, but I won't. I just wanted to tell them all that they've been very, very important to the research over the last 10 years, and why we've been in a position to actually apply for program grant funding and be successful. These are the new group. These photographs are generally taken on the first day, so you can see very happy people, very happy people. But I would think they're still happy. I really would. But very happy people. Jessica runs the lymphoma part of the group brilliantly, and Kieran runs the acute myeloid leukemia familial group. Both excellent clinicians, and both excellent scientists, and people who I think fall into that team that I just feel is so terribly important. I think all the others do and will actually progress to that level, because they're all very talented. Familial leukemia. So Matt Smith is a hematologist here. This is the second part of the story. I remember exactly where I was in the lab when he came. He said, Jude, germline mutation in CBPF. I said, no. Go back and do it again. Matt was fantastic in the lab, very focused. We got a lot of work done. And we published a situation whereby within 2 weeks of each other, a son and a daughter arrived into Bart's clinic with acute myeloid leukemia. We were screening genes. There was only 2 or 3 genes with no mutations at that particular time, and we screened the genes, we found mutations, and we were able to get a publication in the New England Journal of Medicine, which was, amazingly wonderful. What was really nice for me is Jamie, who I see above, and Andrew, Matt. There was never any question whether I'd be last. It was a guarantee. And it could have been so easy to make it a clinical paper. Class people. Other key manuscripts really led because Brian was instrumental in incorporating into the department technology ahead of time. Andrew paid a few pounds to get it in, but Brian set up the technology. He delivered, and what I did, I piggybacked a lot on what he actually achieved. And I don't think I've said it, but I don't think I've ever said it enough. And at the lab meetings, when Brian makes comments in relationship to kind of experiments or genomics, every so often, during one meeting, you'd be going, that's a good point. Excellent scientist. Priya did work on WT1. Karen actually did work on a different WT1 manuscript that was published in the JCO. And what we did was we made our observations based on the tissue bank at Barts. Then we went to the UK MRC clinical trials group. That would be Rosemary and David Lynch. And they provided us with a large panel of patients to actually test the hypothesis that we had defined in the Barts cohort of patients. And lo and behold, we could show the same observation, a link of WT1 with adverse outcome. And fantastic collaborators to have, both short-term and even to this day. Kieran has done an amazing amount of work pulling together these rare pedigrees. And again, this annotated clinical information is just so key and so important. And one of the main observations she's made on familial leukemia, which we'll be publishing quite soon, is that actually clinicians are doing fantastically well because these patients are cured. They're cured, but they're predisposed to getting more mutations, more AML. So essentially, you cure the diagnostic AML, they relapse, and they relapse with a completely new leukemia. And so therefore that changes the dynamic of the disease. And we can actually look in those patients and think about how can we best treat and potentially kind of cure patients, with the suggestion maybe of doing bone marrow transplant in the 50% of patients who relapse. I have to speed up a tad. This is another pedigree, GATA2 pedigree, work by Csaba, who is a Hungarian postdoc who came on a European Hematology Association fellowship. What I want to say about this pedigree is we're doing things that are translational, that are diagnostics, that are making effect on patients' lives. Because this lady, Matt, treated these two first cousins about 12 years ago, and they both died from bone marrow transplant-related issues. The family disappeared, and lo and behold, 10 years later, another first cousin appeared with symptoms. She has 4 young children under the age of 10. We were able to find a mutation, get that information to an accredited laboratory. She wrote an email back to Matt and everybody else saying, "Oh my God, I don't believe it, they're all okay." So her 4 children, we can say, have a wild-type configuration and are not predisposed to getting any malignancy going forward. Good translational diagnostic work, taking the data and information that we're getting through our sequencing information. And that really has led to a fantastic collaboration with Inda Judokal and Tom Voliami at the Blizzard, which I think the LLR can appreciate that we're trying to do something rather special. Because what we want to be able to say in 3 to 5 years' time is that every patient who sits in front of a hematologist, if that hematologist suspects or knows that there's a familial involvement in that pedigree or in that individual, then there's a pipeline that will allow them to get diagnostic testing across the known knowns, about 10 loci at the present time. And that information then tissue banking in relationship to identifying what are the key mutations, the key genes in that pedigree if they don't have a germline mutation. And also to get a flavor of what are the rationale behind the secondary mutations that might be causing inter and intra-clinical diversity within the pedigrees, so that we can define some novel guidelines and improve management. Now, coming to the last part of the talk. For the last 17 years, I've been living a lie, because I've started almost every talk by saying follicular lymphoma is characterized by the 1418 translocation. And now we know through next generation sequencing, this is a 1418 translocation. This is follicular lymphoma. It has these follicles, and this causes upregulation of BCL2, and what you get then is an accumulation of B cells with BCL2 expression. They acquire additional mutations. And the patients have a very kind of odd clinical history in that they're uniquely exquisite to chemotherapy or immunochemotherapy. They go into a relapsing, remitting kind of clinical course and becoming refractory. That's why the median survival is quite good. And we had a game-changing observation going back about 6 or 7 years, because there was a dad who gave his son a bone marrow transplant for acute myeloid leukemia. The son was cured of his acute myeloid leukemia, but 3 years later, dad developed follicular lymphoma. And 11 years later, son developed follicular lymphoma. And so the question is that just, oh my God, this is just an odd occurrence, bad situation? Or is there something that was transferred at the time of the bone marrow transplant that actually led to follicular lymphoma occurring? And that's what we found. It had the same 1418 translocation, the same chromosomal aberrations. And so we could put together a hypothesis. And our hypothesis is that rather than thinking of this relapsing, remitting disease in a linear fashion, and this is well before the solid cancer guys kind of got on board, and we're seeing this across all different cancers at the present time. But there's this pool of cells. These are the cells that we need to kill if we're going to actually affect cure in this indolent disease called follicular lymphoma. The other big advantage about this work is that Manuela came to work in the lab, and I can't say any more. I just think she's the best thing going, and I love her to bits. Now, around this time, I got to talking a lot with Professor Hart, and we used to go to the pub, and Ian Hart's advice was very similar. It would be the same at 6 o'clock as it was at 9 o'clock after 5 or 6 drinks. It was, Jude, get yourself a programme grant, because programme grants for Cancer Research UK were required. And I devised a kind of an idea around personalised epigenetic therapy in lymphoma. It's very important when you're thinking of a programme grant, this just doesn't arrive. This is after 7 or 8 years working in an area where you're beginning to get a flavour of the disease. You're beginning to understand the kind of key observations or the key clinical questions. And that's because you're working with people like Andrew and you're working with clinical fellows in the laboratory. You also need pressure. And this guy can exert a lot of pressure, he can. But pressure is very important. Because if you don't have pressure, you don't push. Nick has been wonderful to me, because what I like about Nick is he tells you what you want. He tells the same thing on Monday as he will on Friday, and he doesn't dress it up. It's black and white. I love that. That's the Irish in me. That's what I really appreciate. There's no second guessing with Nick. You know exactly what you're getting. The next thing you need is 3 professors in hemato-oncology who actually know their stuff. John, excellent at drilling into me from 11 years ago when he arrived. Get the question right. You have to get the question right. You can't have any doubt about why you're doing it, why it's important, where it's going to lead. Andrew has put together a tissue bank of par excellence that's given us an opportunity to be the best in the world in relationship to follicular lymphoma genetics and biology, in relationship to matching up samples with clinical history. And Andrew is there, and he's saying, and you don't forget the pathologists, Andrew Norton and Maria Calaminci. Don't forget the bone marrow expert, I was going to say Mike Jenner and John Amos. And Brian, because Brian introduced technology and a way of thinking into the laboratory that was key in trying to put together those 3 different facets from those 3 individuals into a program that was going to be successful. That is still not going to be enough, because you need 2 more things. First of all, you need to know the game. Louise Jones has been instrumental in helping me move from here to here. Because what Louise did was, when she finished her lectureship here, she made a career change. She worked for Cancer Research UK, and she currently works for the MRC in translational research. But she put my name forward for a grants panel. And so immediately I got a flavour not only of what I needed to do to develop a story, but I also saw how that story was going to be reviewed and critiqued. And then you need 1 more person in the mix. See, short, smaller, see. Heart here. And he cut off his head as well. God, he's so clever. He could do it half his brain. It's great. He taught me how to write. Because you would bound down like a spring lamb, because Ian's a vet, you'd bound down like a spring lamb, expecting Ian to pat you on the head and say, "Go on, son, you're doing..." Ian would say, "Jude, I have no idea what you're talking about." So it's all well and good to have the pressure, to have all these wonderful ideas. But what you need and what part of the puzzle is to actually understand the process, but also be able to write appropriately. And so don't cod yourself in thinking you can be a successful researcher on your own. You can't. It's multidisciplinary. You build friends, you build respect, and you deliver when you get that opportunity. And what I've had is a wonderful team. This is a team of Chaba, Jessica, and Alex. I have a postdoc, a clinical fellow, and a bioinformatician, core funded. You don't do anything on your own. Sequencing of biopsies across the relapsing, remitting nature of the disease. what you get, you can generate a landscape map. And what did we see? These are all individual patients that probably Andrew, Rajneesh, John, everybody has met over the last 25, 30 years that have gone into storage in our liquid nitrogen tanks. Huge cost of liquid nitrogen, tissue banking, and clinical information. But you need it to be able to answer these type of questions. And what do we see? 1418, no more, but a gene called KDM2D, called MLL2, CREB-BP, EZH2, which I want to tell you a little story about. This is an epigenetic disease. There's an addiction in follicular lymphoma. And the three senior authors are the bioinformatician, clinician, and postdoc. Jessica came up with a fantastic way of describing this because we saw two patterns of evolution. We saw something called rich and sparse. And rich is where all the biopsies are pretty similar, but they're more related to this common progenitor pool, the pool of cells we want to kill. But we also see something called sparse evolution, a convergent evolution, whereby what we see here is two biopsies, follicular, transformed, very few mutations in common. But what they have done is they've acquired mutations of a similar type in the same genes. And so in a way they've evolved separately within that individual. And this makes us think, well, you can get some information from the diagnostic sample, but you actually need to be biopsying over time to kind of get a true flavor of the disease. Good. I have 3 more minutes. Another story was we showed Kieran O'Rian. This was a Cambridge Barts grant body that I was very fortunate. Ian and Nick wrote the grant. Well, Ian probably wrote the grant. Nick wrote his name on it. Nick, you're not here, Nick. And nobody's going to tell him. Kieran was wonderful. He did methylation profiling follicular lymphoma, and he found hypermethylation of genes that are linked with something called the PRC2 complex. And EZH2 is one of the key components of that complex, and it leads to a repression. And based on work in Vancouver and us taking on that work, we were able to show that 27% of patients with follicular lymphoma have EZH2 mutations. Immediately you're thinking, EZH2 mutations, gain of function, is there a drug that can target those mutations? And yes, there is. There's 2 different companies at the present time. There's several, but Epizyme is the one that we're actually working with. We're very fortunate. Andy Davis did a PhD with us about 7 or 8 years ago. He came to Atlanta and we met Epizyme. And Andy was brilliant because he said something he said something that I hadn't thought about. But he said something that probably took me 2 or 3 weeks to realize how important what he was suggesting is. Because these are biomarker studies. And how can you use a biomarker personalized therapy when that biomarker might be rare or patients might be cured? And so the suggestion that Andy came up with is that every patient going in for conventional treatment, we should take a biopsy and we should do a mutation analysis. We should define up front whether they have an EZH2 mutation or not. If they have an EZH2 mutation, you can feed that information back to the treating physician so that when those patients relapse, those patients then are appropriate for going into an EZH2 specific trial. And that's something that Peter Johnson, another collaborator and somebody who kind of worked on the CR UK program grant with us, has been formidable in actually putting into action, whereby the clinical trial is now open within the UK, where we're going to be personalizing treatment or seeing whether patients with an EZH2 mutation are more likely to respond. Not in follicular lymphoma, but in a sister disease called diffuse large B-cell lymphoma. And more importantly, there's also now something called Precision Medicine Pipeline with funding from the LLR, another program grant across 6 different centres. It's always nice when Barts comes alphabetically at the top. gives it a little bit more, oomph. Because we're thinking about all these problems. It's very easy to think of personalised therapy. Patient, drug, treat, cure. You have to think of cost assay design, tumor content, tissue quality, the tissue acquisition, the heterogeneity of those samples, the difference between sections and little needle biopsies that Maria has to try and make kind of a diagnostic kind of examination on. But we're moving in the right direction. Across familial leukemia, across follicular lymphoma, we're moving in the right direction. One last slide. Soft Jude could be coming up again. Andrew Lister. Yeah, it just kind of comes in immediately. Tea, milk, no sugar. I have a list that I have to get through every day. And I'm so glad. Every couple of weeks, Andrew pops in. Because Andrew is amazing. A wonderful clinician, cares about the patients, cares about the research. He loves his fellows, loves his clinical fellows. But when he comes in for tea, I can drop everything and we can talk for 10 or 15 minutes. I don't have a list anymore because I'm talking to Andrew. I gave a talk in Istanbul about, 2 or 3 months ago. And it was the best introduction for me because the introduction was Philip Clune. He stood up and he said, and the next talk is from Barts in London on follicular lymphoma. Barts has a huge tradition in follicular lymphoma with Professor Andrew Lister. And today we've got this Jude guy who's going to talk. Honestly, it's the best. To have somebody who you've fought with and argued with, but all trying to move in the right direction, and then to have that introduction. But there's one last thing to finish on. Is there's a meeting in Lugano on lymphoma every 2 years. And I was probably in the lab about 6 months or 9 months. And Andrew said, Jude, you should come to the meeting. Now, I'm pasty white Irish. I don't do hot weather. I really don't. Lugano is a nightmare of a place. Beautiful. It's beautiful. But it's so warm, it's so hot, and it's so miserable. It's so, for me, you walk around, you're sweaty, miserable. Andrew grabbed me. He introduced me to everybody. Because he was yours, and you took care of him, and you took care of the people that worked for you. And you can't imagine how important something like that is. When all your life, that is what's key, taking care of your own. Gentlemen, I'm finished. Thank you all very much.
Well, Professor Fitzgibbon, very difficult to follow that, so I'm going to keep this very short. I'm just going to say thank you. Thank you, first of all, for describing the science, which is fantastic and builds upon a great tradition. But I've got 3 other thank yous to give. One, which relates to your soft side and hard side, is thank you for giving us the insights into your family life, and particularly your parents, and how that's obviously shaped your career and attitude to people, which I think is really important. For the scientists in this audience, thank you for describing those inspirational moments that you just get when you see a southern blot, or you see, in my case, a bacterium growing on a plate, or you understand why you're doing this job and why it means such a lot to you. And then lastly, thank you for your description, I think it's fair to say, of humanity and humility, which describe your interactions with everybody, your mentors, your bosses, and your students, and which have characterized your career. when I was asked if I wouldn't mind standing in for Professor Trembath today, I said, well, could I have a little bit of background information, you know, crack a few jokes about this individual? And it came back with, well, I'm a Fulham supporter. And your description of why teamwork is so important made me think about Fulham. Fulham, for those of you who don't know, club languishing in the lower half of the Championship, used to be a reasonable side, now probably on the slide. But for anybody who is a team player and a team supporter, it's really important that you stick with your team and you always back your team no matter what. I was very surprised you talked about Arsenal rather than Fulham during your description of Fulham. The last thing Jude said was that his one more major regret in life was he didn't drink enough when he was at college. This is what he told me. I think we can rectify that very shortly. But first of all, I would like to thank Jude, and I hope everybody in this audience will join me in thanking him for what I regard as a really exceptional inaugural lecture. Thank you.